So I think it's a very long title, this, but we remain in the field of immunotherapy and antibodies. So let's see. Okay, cancer immunotherapy. Cancer immunotherapies comprises cell therapies and antibodies. We will focus on antibodies and in particular on specific antibody formats. Professor Cattaneo already introduced you to the nanobodies, and we will go deeper in this field. So when we talk about antibodies, we have to talk about antigens. So in our case, we have an antigen, of course, and a recombinant antibody. Which is the antigen? The antigen is a complex, so not just a protein, but a complex between two proteins expressed on the plasma membrane, selectively expressed on the plasma membrane of cancer cells, and the two proteins are represented by an ion channel and a potassium channel, to be precise, hERG. hERG is physiologically expressed in the heart, so many of you know hERG. But the other part of the antigen is represented by an integrin, by an adhesion receptor, and in particular by the beta-1 subunit of adhesion receptors of integrins. As stated before, so this complex is present only in cancer cells as a complex, tightly bound proteins. The antibody is a single-chain bispecific antibody in the format of a diabody. So we started with two monoclonal antibodies, one against hERG and one against the integrin, the beta-1 integrin. Then we produced the CFVs. And then we joined the two. We worked a lot on the linker between the two arms of the antibody to have a true diabody capable of binding only the complex and not the two proteins when they are expressed separately, not as a complex. We published the results of this development. Before publishing, we patent the idea and the product, and the patent is owned by the University of Florence, but was licensed some years ago to MCK Therapeutics, which is a spin-off of the University of Florence, who was in charge of extending the patent, which is now granted in Australia, Canada, China, and so forth. So it is fully protected. And now we come to the title of this presentation and to the work we did on this topic thanks to the financing from the THE, and in particular we belong to the SPOK2 group. And the title states that we can perform immunotherapy in cancer combining the antibody, the diabody I mentioned, with cholesterol-lowering drugs. Why? Which is the rationale of this? The rationale is the fact that the complex, which I mentioned, is selectively present in cholesterol-rich plasma membrane microdomain, so which we usually call lipid rafts, and which are the triton-insoluble part of the plasma membrane. In this specific location, the complex triggers an intracellular signaling pathway which involves classical molecules like PI3 kinase, AKT, RAC1, and so forth, and at the end, it controls cell cycle progression through cyclins and in motility and invasion through the remodeling of the actin cytoskeleton. And so cholesterol-lowering drugs, statins, what do they do in these cells? I'm referring to cancer cells, of course, and in particular to pancreatic cancer cells from here onward. So statins can lower cholesterol in the cells, can dissociate the complex, which is in lipid rafts, and similarly to what exerted by the diabody, by the single-chain diabody. And if we combine the two, the complex is no more in lipid rafts, is no more present in the cells. And the combination of statins with the antibody, which I call here SCDB HERG1 beta 1, let me say just SCDB, the combination of statin with SCDB lowers the proliferation rate and the invasiveness of pancreatic cancer cells. What happens in vivo? In vivo, this is the pancreatic cancer model, an orthotopic xenograft obtained by ultrasound guided injection of human pancreatic cancer cells into the pancreas of mice. You can see here also the images of the ecography, and the combination of statins with the CDB greatest lowers the growth of the tumor and improves the survival. So in the last 2 or 3 slides I showed you, there was this new patent because we have submitted with the University of Florence, thanks to the THE grant, a new patent which has been submitted as PCT and which covers all what I showed you today. And even protected by the patent are these results that are, quite interesting for us. So we show that in the cells, in the pancreatic cancer cells, that the presence of the hERG, of the complex on the plasma membrane, affects the capability of the combination to, in this case, to kill the cells. This is one point. The second point that I think is very interesting is the fact that the combination of statins with the SCDB works even more and is synergistic with low doses of chemotherapeutic drugs, oxaliplatin. So this combination can be proposed to overcome resistance or to be combined since the beginning of treatment with chemotherapeutics like oxaliplatin that for example, in pancreatic cancer is employed as a neoadjuvant, so before surgery. And so we are close to the patients now. What happens in patients, which is the scenario? Here again, pancreatic cancer. The complex is expressed on the tumors in I would say, a high percentage of cases. But what is relevant for us is that it represents a negative prognostic factor. So the survival is worse in patients whose tumor has the complex on the cells, on the tumor cells, and this occurs either in the total population or in some population which are treated with statins for comorbidities. The therapy with statins is very common. So we believe that, we can use statins alone or in combination with the SCDB or other chemotherapeutic drugs for the treatment of patients with pancreatic cancer, especially those who express in their tumors the complex hERG beta-1, because it is a negative prognostic factor, and because the presence of the complex of the channel, makes the combination therapy to be effective, or supposedly makes it to be effective, and this is the title of the patent application which I mentioned before. And last but not least, in the previous slide, I highlighted in red the fact that the combination could be applied not only to pancreatic cancer, but also to other cancers. And indeed, starting from these studies, starting from this patent, what I call a cascade call grant was granted from the University of Florence. The name of the project is RepStat, so it's easy to understand what does it mean. So repurposing statins, and in this case, two companies, GA Lab and AWA Medical, will work together, and will work together also with MCK Therapeutics, because the antibody is produced by MCK Therapeutics, to test the combination in breast cancer, because the rationale, again, is the fact that the hERG beta-1 integrin complex is highly expressed in triple-negative breast cancer. So, thanks to the European community, to the Tuscany, to the university for granting these studies, but the thanks are to Claudia, who is present in the audience, and Jessica, my collaborators and co-inventors of the patent. And thanks to you all for listening to me.
Thank you very much for this very interesting presentation. We have a question here.
Thank you, Anna Rosa. Fantastic talk as usually. I have an idea. You probably know that we have a compound discovered in Florence in phase 1b/2 clinical trials exactly for pancreatic and triple negative, which is a sulfonamide carbonic anhydrase 9 inhibitor. I think your approach with targeting HERG1 statins would benefit from a combination with this type of carbonic anhydrase 9 inhibitor, which is obviously, you know very well, important in hypoxic tumors.
Yes, it's fantastic, and this idea, I would say this was an idea that we already pursued with Claudia, you know, and we worked with you. I don't know whether it is the same compound. Yeah, and Claudia was able with your group to do an ADC, but we applied to kidney cancers. Maybe the possibility of using in pancreatic cancer and triple negative is fantastic. Thank you, Claudia.